The mitochondria, as the powerhouse of the cell, is a common household saying, but it doesn't entirely encompass the amazing work done by the mitochondria. To have an effective conversation about cellular respiration in the mitochondria, we first need to really discuss the electron transport chain, or ETC. Housed in the inner membrane space of the mitochondria, the ETC is a series of 4 or 5 proteins, depending on your professor, and these proteins form complexes that couple redox reactions, creating an electrochemical gradient that leads to the creation of ATP in a complete system named oxidative phosphorylation. Together, these complexes work to complete 1 or 2 tasks, then pass it on to the next complex. The overall goal of the ETC is to produce a hydrogen proton surplus in the intermembrane space and to produce ATP. The mitochondria was keyed as the powerhouse of the cell due to its effective creation of ATP. But more specifically, one complex stands out for its contribution to ATP synthesis, and this is complex 3. It does this through the Q cycle, which is unique to complex 3 due to its novel responsibility of interacting with carrier proteins. These carrier proteins donate and remove electrons from the complex, similar to being passed down an assembly line. The Q cycle is broken down into 2 cycles with 2 pathways for electrons in each cycle. The first cycle includes 1 ubiquinol is reduced to ubiquinone after binding to complex 3. 2 electrons are donated to complex 3, and they are split into 2 pathways. This electron donation promotes the pump of 2 hydrogen protons into the intermembrane space. Our first electron is moved from the complex to the risk center, which is an iron-containing group. This is then passed to one of 2 heme groups inside the complex 3, cytochrome C1. Here, the electron is passed to cytochrome C, causing a conformational change and ejecting the cytochrome C carrier protein to communicate with complex 4. Our second electron goes to the second heme group, cytochrome B, reducing 1 ubiquinone to semiquinone, which is then used for recycling during cycle 2. In conclusion to our first cycle of the Q cycle, we had 2 hydrogen protons pumped into the intermembrane space, we had 1 ubiquinol reduced to ubiquinone, and we had 1 ubiquinone reduced to semiquinone. We also had 1 cytochrome C carrying 1 electron moved off and heading over to complex 4. Cycle 2 is extremely similar to cycle 1. In fact, its pathway 1 is entirely the same, and only pathway 2 differs. In this pathway 1, we see 1 ubiquinol reduced to ubiquinone and 2 hydrogen protons pumped into the intermembrane space. And after releasing a cytochrome C in pathway 1, our second electron is moved to cytochrome B, our second heme group. This is where things begin to differ, because there it is used to further reduce the semiquinone synthesized in cycle 1 to ubiquinol, which can subsequently be reused within the cycle. This reduction of semiquinone to ubiquinol promotes 2 hydrogen protons to pump into the complex itself. In conclusion to cycle 2, we saw 1 ubiquinol reduced to ubiquinone, 2 hydrogen protons pumped into the intermembrane space, and we saw 1 semiquinone reduced into ubiquinol, promoting 2 hydrogen protons pumped into the complex. We also saw 1 cytochrome C carrying 1 electron moved over to complex 4. However, in total, in 1 cycle of the Q cycle, we should probably deduce what we get. We receive 2 ubiquinol oxidized into ubiquinone. Through this process, we have 4 hydrogen protons released, and we have 1 quinone reduced into ubiquinol, which is used for recycling. And finally, we have those 2 cytochrome C are reduced 1 electron each.